Yeah, good. Welcome, dear attendees, to listen to this session on overcoming barriers in pharmaceutical development and manufacturing of nanomedicines, which is a session by the APV, the International Association of Pharmaceutical Technology. We are having a very interesting program, and we can start off with the perspective of the International Association of Pharmaceutical Technology, APV, Professor Jörg Breitkreuz. Please take over.
Yeah, good afternoon, ladies and gentlemen. It's my pleasure to welcome you to our session, I would say, because the International Association of Pharmaceutical Technology has always supported this event for many years. And this year is the first time ever that I'm able to attend your conference. The reason is very simple. In my academic life, I'm more involved in solid drug dosage forms and pediatric medicines, but not in the area of nanomedicines. However, I've been asked by the organizers to present a little bit on the perspectives of nanomedicines for APV. And I'm also happy that Bernd Riebesehl, as our APV contact partner for the CLINEM conference, is also with us today. So I would like to introduce APV first, because we had a very strange history, maybe because we have been founded in 1954 by community pharmacists. Actually, this was after the Second World War in Germany, where there was a lot of missing medicines from industry, and many community pharmacists who built up their pharmacies thought about how to prepare medicines for the patients. Often these have been individualized medicines because they were compounded after the prescription of a doctor. Yeah, but within several years or decades, the focus of APV changed a little bit, and today this association is mainly driven by industrial pharmacists or academic people from pharmaceutical technology or pharmaceutical engineering. So it's well established for many years. It's public and also a non-for-profit association. And I think this is also very important because the community also sees APV as fully independent from pharmaceutical companies, for example, and we often serve as a kind of moderator between companies or between academic institutions and companies if there's a need for this. So today, we are one of the most important international societies in the field of industrial pharmacy and pharmaceutical technology, with about 1,700 members, and about 1/3 of these members are non-German citizens located somewhere else, mainly in Europe, but in total from over 30 countries. What is also quite unique, I would say, we have a database of real 18,000 addresses of in-job pharmacists in companies and also academia, which sometimes gives us good opportunities to spread our good news. So this is our APV board, and you see that we try to cover almost all areas of industrial pharmacy, also with the members here of our board. You see myself, and then Heribert Häusler, who is our vice president. Formerly, he was the head of quality within the Boehringer Ingelheim company. Then we have our treasurer, Katrin Bartscher, who is the chief scientific officer of Next Pharma, so CDMO. We have Caroline Bechtold-Peters, and many of you in Basel may know her because she's with Novartis, and she's mainly in the field of biotech drugs, and therefore also in the arena of nanomedicines. We have Johannes Bartholomeus, formerly the head of development of Grünenthal and now driving his own company. Professor Sandra Klein from University of Greifswald, and Hans Lindner, formerly for many years with Bayer, also in the area of nanomedicines, but today he is the CEO of Nuvisan, also a CDMO. And we have the senior vice president of Grünenthal, Martin Lück on board. So this is our board where we take most of the decisions on behalf of APV. And you will also see that we have a quite strong office. Our headquarters is located in Mainz, Germany, where APV was also founded. And maybe the most important contact partners you might have are our managing directors for our association, APV, EV, but we also have a profit organization, which is a GmbH, and here the managing director is Katrin Kerpert. And if you send out some communication by email or so, you might also get into contact with the other persons. So what are our topics? I said we try to cover almost all areas of pharmaceutical industry, and therefore you find a lot of typical business and typical positions also on the right-hand side: drug regulatory affairs, quality control and analytics, information technology, quite much rising part here because of GAMP discussions and so on, engineering, quality assurance. We have some events on management tools and self-education, I would say. We have the pharmaceutical biotechnology. I will comment on this later on. Pharmaceutical manufacturing, research and development. And sometimes we pick up hot topics to be very quick with some new events, and we can really organize such an event in a few weeks or months. So then we are organized in so-called focus groups and also an increasing number of so-called task forces. First of all, what is a focus group? A focus group is a group which continuously works over the years. And we established such a focus group with dedicated members who are in this field, and then these members of the focus group can decide whether they want to talk in German or English. You see the German-speaking focus groups on the left-hand side and the fully English-speaking groups on the right-hand side. And in bold, I pointed here those focus groups where I think if you are working in the field of nanomedicine, this could be interesting for you. This is a drug delivery focus group where Bernd Riebesehl is a member of, and there's a pharmaceutical biotechnology part. And if you are interested in some of the others, just let me know. Here, for such a focus group, the APV board appoints the members, which means that there's a certain budget per member which is available for meetings and so on and so forth. This is a big difference to our other groups, the task forces, which I will show later on. So first of all, the focus group drug delivery. The mission statement of the group is to combine science and technology in order to create advanced drug delivery systems. And I could imagine that most of you as attendees here of this conference would summarize yourself in such a way and in this field. And here you find an old picture of Bernd. Sorry for that, but I took it from our homepage. And here are some other members, and there are even some members missing, those of this group who did not want to show up by a picture on the website. Yeah. And here you see an important thing, of an important output of this group. They are producing newsletters with really nice contributions on very new products. You see, for example, last year, in the beginning of last year, they already had contributions in detail for the BioNTech vaccine, Comirnaty, and some others as well from Moderna and so on and so forth. So very hot topics, very close to the recent investigations, and this can be sent out as a freely available newsletter after you register once. So everyone can take it for free. Very important contribution of this group. Here, as further examples, you see the next newsletters. So if new drug delivery products come out, then usually it takes only a few weeks or months that you have a detailed analysis of them and really introduction to the products. Drug delivery companies, here you see Precision Nanosystems from Vancouver, and you will see in our session today a speaker from this company. So we are trying really to be up to date there. And sometimes we introduce here drug delivery people who are of interest for the community and literature and so on and so forth. Here on the right hand side, you see the next one, which was from December last year. And here you see also some other information, and we are looking forward for the upcoming newsletter. I think it will be in this month or next month, the next one. Okay, then there's another pharmaceutical biotech, then another focus group, which is pharmaceutical biotechnology, which can be of interest. They are not releasing such a newsletter like the drug delivery focus group, but they have very interesting seminars where you can attend and meet the people in charge. So this group was founded by Hermann Allgeier, who was formerly the CEO of Merckle Biotech and was very well known in the community. Caroline Bechtold-Peters from Novartis was mentioned earlier, but we also have some more people. Maybe some remarks. We even have contacts to the EU Commission, because Heiko van der Leyen is the husband of our European Commissioner Ursula von der Leyen. So you see that there are even direct links to politicians here. Okay, I would like to emphasize that there will be a very interesting APV workshop on protein aggregation and immunogenicity very soon in Basel, Switzerland. So exactly where you meet today, if you would be there in person. And this conference has been founded actually in the US some years ago and was quite well known there as a Breckenridge conference in Colorado. And now frequently they support us here as speakers and bring you the newest information in this area also here to Germany or Switzerland. Okay, then we have the task forces, and there's a difference to the focus groups, because the task forces are proposed or can be proposed or spontaneously formed just by groups of our members or stakeholders. And you see yourself here the topics which are quite hot in these days. And I would like to stress here, there are 3 topics which could be interesting for yourself, which is aseptic in German, but individualized medicines and nanoformulations as well. I would be also very happy if this nanoformulation group would be a little bit revived by some new interested persons.
1 more minute.
Yes, then we will have, or we have the world meeting, which is quite well known, where we propose you to come, because there are many topics also on nanomedicines. This year it was in Rotterdam, 2024 it will be in Vienna, Austria. And last but not least, in the uneven years, we also founded a European conference with our partners from Italy and France. And next year there will be the meeting in Marseille, France, and we have a lot of talks already invited, which are in the field of nanomedicine. And I would be really happy to meet you there. So as my time is over, I would like to say that please enjoy this session and the entire conference, and hopefully we can see in person at one of our events or next time at the Cleanup if I can attend. Thank you very much and enjoy the conference.
Thank you very much. Do we have questions? Let me check here on the debate channel. No question yet. So let's see again. Okay, we don't have questions on this. Thank you very much for this overview, APV. And now we switch the next speaker. So it's my pleasure to announce Kurt Sedo, Vice President Operations from Pharmaceutical in California, to inform us about the changes in nanomedicine research learned from the evolution of global pipelines and markets. I think we have seen quite a change in direction on the nanoparticle side, and so I'm very curious to hear from Kurt what the market insights are. Please, the stage is yours.
Great. Thank you, Bernd. Thank you, everyone, for your attention. Yeah, I was going to be looking at giving an overview of the industry here and some trends, some overall trends, and then kind of narrow back into some of the nanomedicines and nanotechnology and some future trends where, nanoparticle, nanomedicine, nanotechnology can be incorporated into the drug discovery aspects as well as the delivery aspects of pharma. So we're looking at here, just kind of looking at the industry as a whole. These are NME and BLA approvals through CDER here in the United States. So the pipelines are continuing to grow. There still are new medicines being developed. Biologics are a big part of that, but as we're going to see here in some of the other slides, small molecules have not gone away as in matters of importance and as matters of being developed. And a lot of those are being discovered utilizing artificial intelligence process, nano discovery technology platforms. So there's a lot of ways in which nanotechnology can be incorporated into the entire process of developing new pharmaceuticals, either in the formulation end, the discovery end, or in the active ingredient end. So there's lots of ways in which this nanotechnology can be incorporated into these new products. But as far as an industry, it's going very, very well. And I always look at sales, seeing what these product sales are. It was really interesting. If you were looking at the 2010s to 20s, you would thought that the biologic space would be completely taking over. But with the new modalities in drug discovery, we're seeing some very, very, very good small molecule drugs being into the market. And Eliquis, 17 billion in sales last year. That's a very, very, very large number on a small molecule. Revlimid, Imbruvica, Xarelto, Brictavri, those are all small molecules. So it's really looking at not the resurgence, but I think the importance of looking at all modalities for looking at new treatments, whether it be cell therapies or gene therapies, traditional small molecules. All of these have a place in developing these new medicines. Now we're looking here kind of at the pipeline. I'm looking at innovator pipelines here and looking at here by molecule types. Over the last 6 years, we are seeing incredible growth in numbers of companies, numbers of programs in preclinical to clinical stages. This represents everything from the discovery preclinical stage on through registration, because then marketed, we could do a different analyses. What we're seeing is a lot of growth in what I'm calling advanced biologics. It's really looking at nucleic acids, cell-based therapies. So the nucleic acids, you're looking at the different RNA structures, constructs, certainly gene therapies, then the cell therapies, and then also gene-based cell therapies, where you're manipulating the actual the cell construct and reintroducing that into the human body. So we're finding those types of things. The cell-based are really early stage. A lot of it's preclinical research. I don't have that data here, but we could talk about that in the breakout session if we wish. Looking at it by a route, injection is the overall route. It's driven a lot by biologics, but it's also driven a lot by the small molecule drugs. Or really cancer is the indication. So those tend to be injectable. So injection is really taking over the route of administration for all these new drugs. And that's even becoming true in the approved drugs. This year, I believe the FDA innovator drugs, the injectable drugs was more than oral drugs in new novel NDAs, not just NMEs, new formulations. The injections are really taking over in all aspects I'll show you later too, in patents and drug delivery technologies and in products. It's really becoming the driver of the industry. And looking at here, pipeline, these all match, showing the immense growth in the industry, looking at the indications, cancer, infections, CNS, endocrine metabolism, really, really driven mostly by diabetes. And then I would say inflammation and immune is looking at these different disease areas versus year. Again, steady growth across. Again, you're seeing these things matching up with small molecule injectable. So you're getting kind of the picture that it's injection, cancer, infections, CNS. To show you the CNS is now being treated with these long-acting drugs. So I'm going to highlight some of those technologies with some very rudimentary nanotechnology, but still, those products would not be enabled without those techs. Also, kind of look at idea where the money's going. This is always a good idea of where the investments are being made. So this is by disease area, and one thing that really struck me was in the genetics. There's going to be genetic disorders, things like Duchenne's and Fabry, these rare diseases, in diagnostics. So that whole information here, this segment of diagnostics, analysis on a chip, diagnostic directed therapies. Again, those are becoming really part of the industry now. And I have some slides and some thoughts on that as well. Look at some recent approvals here. Here, looking at it, this long-acting injectables. I mean, the nanotechnology here is certainly not anything earth-shattering. This technology has been around for a long time, but without that nano milled-enabled formulations, you wouldn't get the release rates out of here. It's a release rate driver, but it enables these long-acting drugs. So in the case here, the Invega half year, it's a 6-month schizophrenia treatment. That's a game changer in CNS. Now we're looking at HIV. These long-acting injectables are going to really change the treatment of HIV in many, many, many regions. Looking at a 2-month dosing schedule, that's really, really incredible for a disease that needs to be monitored, and you don't want to skip your doses, things like this one. And some recent approvals here, just for interesting things. This is this Skytrofa. It's a long-acting HGH, long-acting from Ascendis, a really cool technology. It's a new novel pegylation technology. Basically, you cleave the peg off without having a residual linker, so it's really has greater activity. It's a next generation tech, and some interesting approaches on looking at repurposing old drugs. That's still not going away. Like I think in the last slide, we're showing that the FIARO, the albumin-bound sirolimus, that's a nanoparticle technology on an old drug. So there are still opportunities to repurpose and remake these old drugs better with nanotechnology. That's certainly there. And I think this treatment, this is an area that has been really hasn't been advanced as fast as I thought. Now this last year, 2 groundbreaking products, a contact lens with a drug, a refillable implant for Lucentis, so 6 months. Those are very, very, very important technologies in looking at diseases. This is the last slide I'm going to show you on this risperidone franchise. It's been a favorite of mine for years. This is really how you take a really good drug and continually reformulate it to meet the market needs. With I think the penultimate product here on this franchise is a 6-month injectable, which is a nanomilled formulation. So I'm going to slip into the drug delivery technologies here. Now, here at Pharmaceutical, one of the things we do is we track the drug delivery technologies that are in the industry. So kicked out from the venture phase up through the large pharma. And we're seeing here again, it's mimicking what is being done in the pipeline and products. So as those pipeline and products come around, the technologies being developed to deliver these novel constructs is really going to be mirror the route. So the injection, and that's going to include formulation technologies, lipid particles, liposomes, all those types of things, as well as devices. So here we're looking at I'm going to overview routes on injections, really devices, and biologics that are non-cancerous. So for anything that's inflammation immune or migraine or any of those things, it's going to involve some sort of a device. So we're looking here infusion. We're seeing a lot of switch from IV to sub-Q. So we're seeing more sub-Q products in the clinic, and then we're seeing IV sub-Q switches in the marketed products. We're seeing several technologies out there for monoclonal antibodies switching into sub-Q with large volume injections and site absorption enhancers and things along that line. Also with the devices, we're seeing the device constructs here being compressed. Looking at examples of Enbrel, one of the the early biologics, really took over 10 years, almost 10 years to develop a whole suite of injection devices. In the current market here, we're looking at multiple launch approaches, and a year later with another device. So they're coming right out with a novel entity and a novel device. And the biosimilars are following the same pattern. So we're seeing companies like Mylan and Biocon coming out with innovator or biosimilars of things like Humira already in a pen and an auto injector. That's a little bit of the differences of nanoparticles, but it's important to understand that segment of the industry. In a place that Bernd and I and others are really interested in is these long-acting injectables, and it's by design. So you can modify the active construct like an FC fusion protein. You can do like with Victoza, you're putting on a tag that binds to albumin circulating in vivo, extending the half-life of the GLP-1 analog. Then we're looking at depot, which is going to be the things you're actually formulating into typically PLGA or some other polyethyl polymers to get a long acting, and then PEG polymer conjugates, things like Neulasta, Integrity, and Trefoya. Those particular things are again looking at extending the half-lives of these medicines and decreasing the frequency of administration. Again, the industry is really starting to put some steam behind this, so this is something to look out for. Overview on some routes here. Here's some basics. Oral still the preferred route, and that's really easy to do. It's even though injectables are the leading the pipeline, orals obviously has its advantages. Nasal's got some interest. Inhalation It's small but steady. Ophthalmic, we're starting to finally see some really game-changing type looks here into that space here. Topical, it's steady. Transdermal is pretty much really flat or almost non-existent. The new molecules being discovered are really they're just too big, and they have some lipophilicity issues and things along those lines that really don't make them amenable to transdermal. Possibly opportunities in microneedle, possibly opportunities in vaccine delivery via microneedles. That's an opportunity there. Here are just some notable approvals here. Again, there's other big presentations showing these things. Kind of show in the nanomedicine spaces things that have done along the lines here. Some of the newer ones here, we got like liposomes that we're looking at the first, on patch with the first cationic lipid-based systems. So inhalable liposomes is going to be a first treatment here now, delivered to the lung for a very, very difficult disease to treat. Recent approval. We're looking at nanoparticle formulations for eye diseases. Again, once and twice a day versus 4 times a day is a big difference in trying to apply these drops to your eye. And then also nanomilled suspensions for IV. Here we're looking at meloxicam, a non-opioid medication. Again, the opioid crisis is now looking at repurposing some of these other drugs to treat these pain indications without opioids. In this instance here, you've got a nanomilled suspension. Again, nothing earth-shattering on this technology. It's the nanocrystal tech, great technology. It's been around for 20, 30 years now. Again, here the vaccines, which were completely wonderful addition to the human disease treatment. These technologies enabled these vaccines to be developed. It's not possible to deliver these without those technologies, and that's the forefront now of the nanomedicines. In these products here, really is regaining confidence. We're seeing people going, these are real technologies. We can make value-added products on these things. These aren't just dreams anymore. These are real things. So people are beginning to look at this again and be willing to take some more risks on these, because those kind of went a little bit shy. People were shy to invest in these particular pieces. And I'm looking here, and this is from data from our system, some of the injectable nanoparticle programs by phase. Again, a huge stack of these in the preclinical. That really hasn't changed. We're seeing a lot of buildup in preclinical, and then either phase 1, either they don't pan out, or they don't make it to phase 1 for a variety of reasons. And I think a lot of them is funding and things along those lines. Technologies within our system, we're tracking technologies from, again, industry here. We're looking at polymer, we're looking at lipid cationics, still a lot of liposomes. A lot of liposomes are using cationic polymers. Again, we're going to be delivering either cancer agents or looking, again, also delivering nucleic acid constructs. Here, patents, again, we're covering those. There's continued patent growth over the years. They're still not going away, and this is drug delivery. We're seeing that increase. We're seeing that similar type of increases in publications in nanoparticles. So this is very, very much alive in the early stage. It's now, how can we continue to move these forward into the clinic? Just some future thoughts here, and we're looking at CDMOs are again part of the drug development, the drug delivery, the discovery, the manufacture. Diagnostics are a big part of treatment. They're going to guide treatments, and there's nanotechnology in these diagnostics. Artificial intelligence, machine learning, digital data, all of these things are being utilized to in the early stage discovery. And now we need to put that into in my particular interest is in the drug delivery and how can we utilize these. We have digital therapies, so the non-medical treatments for treatments, especially CNS, ADHD, anxiety, depression. These beyond apps, they're actually really, truly clinically validated pieces. Also, medical devices not using drug therapies. So electrostimulation for CNS and medical diseases. We're starting to see companies looking at that, or natural therapies, cannabinoid, mushroom-based, psilocybin-based entities that are going to be looking for those particular pieces here. So in conclusion here, we're looking again towards injectables, biologics, and cancer. Small molecules are still the largest. Development of advanced biologics, we're starting to see people push those forward, getting out of the clinic, getting out of preclinical, getting in the clinic. CDMOs are a big part. We have lots of investments in all areas. Diagnostics are integrated. We have machine learning. We have digital therapies and medical devices as non-treatments. So with that, I'd like to thank Dr. Tudor Corrali, our president and founder, Dr. Shayo Tukin, our director of drug delivery, and Selva Jandan. She is our director of vice president of data and analytics. Thank you very much. And we have some time for questions. Looks like 1 minute.
Yeah, thank you very much, Kurt, for this very interesting overview on pipeline trends and market. Let me see whether any questions were submitted. Please submit your questions in that box if we have, if you want to take more time, we will have later on the session with the debate lounge. Later on, we all see a link that you should click to switch over to that lounge so that we have an unrecorded session for exchanging. So let me see. No, I have no question. Okay. One more trial. So I need to update again. Question viewer. No, no questions. So let me see. Okay, then I thank you very much, Kurt, and please, all of the speakers switch over to the debate lounge later on so that we can answer any upcoming questions also later on. Now it's my pleasure to announce Professor Mark Chiu, President at Tabotec Biotherapeutics at Malvern, Pennsylvania, to talk about novel human single-domain antibody-drug conjugates for oncology. The stage is yours, Mark.
Thank you very much for the kind invitation. And I guess I'm following Dr. Sato in the sense that one of the great opportunities lies on mixing and matching the different types of modalities. And that's what I'm going to do today, about how one can improve upon directing therapeutics based on homing, increasing the specificity, potency, and ultimately the efficacy. So in some regards, one of the great opportunities in drug discovery lies in going beyond the targeting on the extracellular side of the proteins and trying to have higher specificity with regards to controlling the intracellular target space. I mean, a lot of the things that we do, we heard lately that there's a lot of these small molecule efforts that are taking place that represent one of the largest distributions of drugs that are in the market right now. But a lot of opportunities in terms of going beyond the proteins, in terms of the nucleic acid modulations, the degradation of disease states that are dysregulated, and making sure that we understand well what we want to more or less shoot for. So in some regards, there are imperfections, there are challenges with each one of the targeting modalities. For small molecules, systemic distribution works well, but with the specificity and challenges with ADME, you do have a lot of side effects that comes up. With antibodies, you do have the specificity, but you don't have the intracellular targeting because of the relative size of that system. Mixing and matching the two, you still have to work on improving the internalization inside the disease areas of interest. And people have been experiencing different variations of PROTACs by which you can have more specific intracellular targeting, whether you then use that as a small molecule based in PROTAC or trying to use genetic methods or even with cells. A lot of the still the issues of how to home into the selective disease area of interest. And because of these challenges, this is that lack of differentiation between disease cells and normal cells. You don't have a broad therapeutic index. So if you can more or less solve this problem, you can help a lot of patients who suffer from this inability of getting good treatments. So in terms of trying to target intracellular target interactions, one of the great challenges lies on lack of interacting surface area. So people have been exploring, looking at peptides, having peptides that have an intrinsic length of anywhere between 3 and 20 amino acids, more surface area to have that engagement, to have specificity, but more or less trying to capture this in a more structured presentation. That structured nature of the peptides is going to be important to increase the specificity as this biotechnology company has explored. Now, when you look at the literature with this, there are challenges that comes up in terms of looking at their preclinical data. lots of opportunities in which they can control cell viability or mediate tumor killing, but still with a lot of the cyclic peptide presentations, the challenge still is the lack of potency, which means that you have to have high dosing levels. And because of the nature of the presentation of these peptides, you do have to worry about pharmacokinetics. So you don't have enough of a dose response that means that you have the frequent dosing. So trying to solve this is something that we wanted to approach. Now, one of the things that we're trying to kind of focus on is the ability of using our toolkit, the Tavo Elite system, in which what we're trying to do is increase the specificity to the disease tissue area. And this is more or less focusing on particular markers on the disease cells that are not found in the normal tissue area. Having the intracellular targeting that uses a presentation of multiple cyclic peptides to mediate the specificity and the potency. But this means that you do have to control how you have the penetration within that cell. So once we have that homing internalization component, the focus on how we engineer that so that allows us to neutralize the target, preventing the protein-protein DNA interaction, and then thereby allow for the targeted destruction using particular component domains. So now, if we're going to be working on a system that allows us to have more specificity, but also have the constraints of having a smaller size, what we wanted to do was look at a nature of a cyclic peptide presentation. You know about that in the case of antibodies. We were looking at much more smaller single domain based human sequence molecules that allows us to generate a presentation of these different cyclic peptides. Where you have an antibody of a particular size, we can drive this by having these single domain human variable heavy chain only molecules of a much smaller molecular weight. And then we can introduce different kinds of components to increase the PK, which allows us to have a molecule that can range between 35 and 74 kilodaltons. This selection of a single chain format does allow us to more easily prepare this using the conventional GMP CMC based methodology. But having that smaller VHO allows us to penetrate through the diffusion barriers that different tissues may have, and allows us, of course, to be able to link these together to increase the avidity in terms of targeting our molecule. So we have a phage selection pathway in which we can generate different epitopes into this particular VHO format. And that choice of how we do the selection allows us to guide whether you want to have IV, sub-Q, oral, pulmonary, topical peptide delivery based systems. Now, the basic systems that we have is a self-assembling system that we put together where we have the ability to target more than one surface antigen that by specific nature increases the specificity in terms of homing into disease tissue. We have structural components that allow us to have the internalization that goes into the endosome with some components that allow for the endosomal escape so that you can now have the presentation of the VHOs to go towards the cytosolic target to allow for inhibition and degradation. So we were able to generate a wide variety of molecules. And one of the themes that we do focus on is making sure that we have cross-reactivity in terms of the target we have of interest, both for advancing into animal models as well as getting into relevant toxicology models so we can transition things into the IND sooner. When we make these particular molecules, they're easy to generate in terms of the purity and looking at the relative aggregation state, which more or less leads into being able to use the standardized CMC methods of generating biologics, antibody-based molecules. So Just an example in terms of how we want to look at this. We do have that ability to engineer the particular targeting antigen on cancer cells and generating the different types of specificity molecules. We can engage that different types of tumor cell engagement to allow us to understand that we have that homing to the cancer cells themselves. Now, one of the other aspects that we do look at is once you do have that target engagement, we do spend the time to look at the internalization potential, that nature of the VHO that allows for that epitope engagement so that you would have that internalization are some aspects that we go beyond just looking at the binding to the cells or binding to their respective targets. And because of the modular nature of the single domain system, you're able to do experiments in which you can vary the orientation of the different molecules, where you have the 2 molecule ahead of the 13 or the 13 ahead of the 2. Different variations of these tandem sequences will result in different types of activity. So looking at the intrinsic property of internalization, we also do add different types of domains that we can introduce so that we can have higher levels of internalization of these molecules. And that exercise of empirically changing the different orientations, the linkers, the architecture of each one of the components will allow us to have increased levels of internalization. Now that we have the homing component, the internalization component, we have to have another component that allows us to bind to the intracellular target. And we could do this by doing an ELISA-based response. And we can see that at different concentrations of these biologics, we can in fact have engagement. And you can more or less screen for engagement of the less active and the more activated forms of the intracellular target. And of course, we do focus on having a target that is a key component in terms of the pathway regulation of increased cancer cell growth. So we have identified a biologic that can bind the activated form better than the inactive form to more or less increase that potential we have of a more specific molecule for the disease state. Now that we have this aspect of the binding, the next thing we do is we try to look for the downstream signaling response. So in this particular case, we focus on a biologic that is able to target a particular intracellular target. It is well known to have induction of subsequent downstream signaling cascade. So I showed you earlier the ability by which we can have the engagement onto the target in vitro. We can then also allow for the intracellular delivery into 2 different non-small cell lung cancer cells. And that engagement of that receptor means that we see the decrease in the phosphorylation downstream of that particular receptor. I show this type of a assay to show that we have that specificity in terms of engagement, because targeting other related signaling cascades may lead to undesired toxicity that comes up with the lack of specificity in terms of that signaling cascade. So particular examples of 2 small cell lung cancers are being extended into different animal models at the moment. We have been looking at target degradation because there are instances by which inhibition is insufficient because of the feedback loops that take place within the intracellular targeting pathway. So in this particular example, we can demonstrate that we can get a molecule that can get inside of the cell. And if you add one particular component, you can then allow for targeted destruction of that intracellular molecule. So we're more or less taking this toolkit to now put together this Tableau Elite-based system that more or less pulls together all the different BHO components that allows us to have homing to the tumor cells, control regulation and internalization of that molecule. And then we have the choice of having inhibition of protein-protein interactions inside. You can also have that ability of degradation also. We are expanding our efforts in terms of being able to deliver different classes of nucleic acids. So far, we have been using the FC domain as a way of controlling the self-assembly and being able to have the tunable PK. And that more or less allowing us to have a molecule that gets as small as 35 kilodaltons, and the additional molecular weight could also be used to have more specificity in terms of engagement. And it really depends on the type of cancer cells that you want to work with. So in conclusion, I'll give you an example of modular components that allow us to find unique architecture to more or less have a much more specific homing, internalization, inhibition, and targeted destruction. So with this, I thank you for your attention. Sorry I'm not able to be there in person, but I look forward to that opportunity to speak more with you. So thank you very much.
Yeah, thank you very much, Mark. I just check whether we have any questions in the chat here directly. No, that's not the case. So let's have all other open questions in the debate lounge. And now we switch over to the next talk. I'm inviting Dr. Lloyd Jeffs, Senior Director of Biopharmaceutical Development at Precision Nanosystems in Vancouver, to talk about the accelerated development and clinical readiness of genomic medicines.
All right. Well, thank you very much for the invitation.
The stage is yours.
Hello, can everyone hear me?
We hear you. Yeah.
Great. Thank you very much for the invitation today. Very excited to speak about the work the Precision Nanosystems team is doing and has been doing over the last little while. As many of you may know, I'm trying to advance the slide. Precision Nanosystems is a global leader in nanoparticle solutions, working with our clients and our partners to provide innovative solutions and end-to-end nanoparticle projects. So we have worked with on over 150 projects, and our nanoassembler system for making nanoparticles, we've placed over 700 of those around the world. So we're very excited about that. And genomic medicines are the future, and they do treat diseases at the fundamental level, either to silence genes, express genes, or edit genes. And we can target any gene anyway. So that's the promise. To unlock the full potential of genomic medicines, we need to look at two things. One is the economic challenge, and one is the delivery challenge. Basically, for delivery, it's getting the active pharmaceutical ingredient, which is often a large RNA, to the right cell at the right site of disease. So we need to really understand and design our nanoparticles and nanomedicines to do their job, whether it's through intravenous route, intramuscular, nasal, transdermal, or even through the topical routes. So lipid nanoparticles, as we've seen now, with over 11 billion people receiving the mRNA LNP vaccines made by Pfizer, BioNTech, and Moderna, they are a safe and effective means to protect an RNA drug substance and mediate RNA drug delivery into the cytoplasm where it's active. And Precision Nanosystems has been founded on basically microfluidics technology to precisely control the formation of the nanoparticles that encapsulate their payload. And we do this using our next-gen microfluidics technology, and it's very scalable. And we can do nanoparticles, we can do polymeric nanoparticles, exosomes, liposomes, and emulsions using our microfluidics technology. So this uniform scalability of nanoparticles using microfluidics is based on this time invariant mixing, where the mixing and particle formation characteristics are consistent over time. And that gives us very reproducible formulations and very uniform formulations. You can also make nanoparticles using turbulent approaches like tea mixing or impinge inject mixing, but inherently they're turbulent and require additional process development time and effort to figure out the critical process parameters and mixing conditions. You can also, with our microfluidics technology, scale up by using larger mixers and also scale out by doing those in parallel. So we provide scalable solutions from research through to commercial. So helping people, scientists with discoveries and helping them scale up their nanomedicines all the way through to commercial using our nanoassembler systems and the consumables, the mixers. So you can go on a Spark with under 1 milliliter batches with our commercial system, which we are currently developing, that can go up to almost over 1 liter per minute. Or 60 liters per hour. And here's a little bit of data. This is for a lipid nanoparticle that contains EPO mRNA. And we see, regardless of the instrument or the scale, we get very similar particle properties with very small polydispersities, indicated on the left by the black circles, and very high levels of encapsulation efficiency. So that scales over all of our instruments. So that makes drug development much easier for the researcher. Also, now Precision Nanosystems is part of a larger company. Danaher is our parent company, and with our sister operating companies, such as Aldevron and Cytiva and PAL, we can provide an end-to-end manufacturing solution. So for our nanoassembler instruments, they have been installed both at client GMP sites and also at CMOs and CDMOs. And there's no technology access fee or royalties associated with our instruments. So from the very beginning, you can work with Aldevron to make the plasmid DNA. It's the basis for the in vitro transcription reaction to manufacture messenger RNA and self-amplifying RNA, particle formation with the nanoassembler instruments, and then downstream processing using tangential flow filtration, sterile filtration, and fill finish using Cytiva and PAL solutions. So talk a little bit about our lipid offering, Genvoy, and it's a proprietary state-of-the-art technology, and there's intellectual property associated with that. And we've seen that novel compositions are designed to optimize of established diseases or use cases such as vaccines, gene therapy, and cell therapy, as well as other emerging applications. So we have a library of novel ionizable lipids and formulations, and we can design formulations for the need. And here's an example here of the Genvoy ILM, our reagent, showing that it can effectively deliver in vivo both messenger RNA and self-amplifying RNA, resulting in protein expression. And here we're delivering a F-Luc messenger RNA or self-amplifying RNA, where we see good expression, both with Genvoy and a published formulation based on the ionizable lipid MC3. You do see different profiles of protein expression with self-amplifying RNA. And in this experiment, self-amplifying RNA was at 1 microgram per leg, where messenger RNA was a higher dose of 5 micrograms. So RNA formulation and process development, this is key. So we need to basically understand the critical quality attributes of our nanomedicines and also understand the process development, critical process parameters. And we typically do that using a multivariate design of experiments approach, and that provides insights and allows for the acceleration of formulation and process development. For transitioning from discovery to preclinical, we obviously look at RNA LNP formulation characteristics, particle formation parameters, downstream processing conditions, such as buffers, the type of TFF system you would use, and sterile filtration. But critical to all of this development is analytics. You need reliable and informative analytical methods to make decisions on your formulation. And that'll help you develop target specifications as well. And as part of the analytics is a biological analysis as well. So that could be bioanalytical work in vitro, but also the critical in vivo data to look at the toxicity and PK/PD. And I stress this to everyone, if you're going to make a formulation, even if it's your first one, keep some aside and put it on stability. That will be very, very useful. You cannot make time. And the sooner you get your formulation on stability, the sooner you'll have meaningful data on the shelf life of your product. So as people have become appreciative of mRNA LNP vaccines are complex drugs. You have to look at the lipid composition and the RNA quality, also the payload design and some of the where your drug is inside your formulation, the encapsulation efficiency. So we've concentrated on looking at the RNA integrity and also looking at the lipid chemistry or the analysis of the lipids. So we characterize, look at, in this case, self-amplifying RNA integrity using capillary electrophoresis. And we've seen that under certain conditions, LNP containing RNA sometimes involves high shear forces, low pH, higher temperatures that can result in the degradation of the RNA. So we monitor that very closely as we develop our process. Also using lipid analysis can identify process issues. Here, a standard tool is UPLC with a PHAD detector. And it's essential to assure that your composition is maintained throughout the formulation process and the ratio of your lipids to your drug is maintained as well. And here you see a 4-lipid component system where we are able to resolve each of the lipid peaks. Okay, so this is quite a busy slide, but I do want to kind of highlight going from development and tech transfer to GMP manufacturing. It's in two parts, the preclinical and clinical development that typically takes place with the client, and then the CMO activities that will, where tech transfer to a GMP manufacturing site or a CMO is critical. We know for technology transfer, transferring processes, materials, equipment, and analytical methods need to be very precise, and you need to transfer everything and understand the responsibilities. You need everything in place to successfully manufacture and release a GMP batch. So just the last part of my talk, I do want to talk a little bit about the our P&I's support from the Canadian government through Canada Strategic Innovation Fund in two parts. One is to develop a COVID-19 vaccine using self-amplifying RNA at P&I, so the government is our partner. And the second funding we have received is from the for a genomic medicine manufacturing center that has been supported by the Canadian government, which will be opening in 2023. So if you're in Vancouver next year, we would love to give you a tour of our new facility. Our new facility will have 2 drug product manufacturing lines, and we're very excited about that. For our COVID-19 vaccine program, we are ready to manufacture GMP batches. We've done tech transfer to a local CMO, and we have identified our GMP drug substance, and we've made engineering batches. So we're ready to go and start a clinical trial later this year. Why self-amplifying RNA? Well, potentially it's 10 to 100 times more potent than mRNA vaccines, because it will basically self-amplifies after injection inside the target cell, and it will allow for basically a larger expression at a lower dose. So that hopefully the potential would be to reduce doses by up to a factor of 100, and also reducing manufacturing burden, which is important for rapid responses to pandemics. As we've also seen with this formulation, that we get very reproducible particle size and encapsulation properties throughout scale up, which is very helpful and predictive. You see 2 formulations here. And we've also conducted in vivo testing of these self-amplifying RNAs, encoding the pre-fusion stabilized SARS-CoV-2 spike protein, where we inject prime at day 0 and boost at 4 weeks, and take serum samples at both times and spleen at the end. And we've seen in vitro activity of both of the formulations I see shown here. It's both efficiently induce SARS-CoV-2 specific IgG response. We also observed that in vitro, so the in vitro data is predictive, and generated neutralized antibodies against SARS-CoV-2 virus, and both formulations showed effective cellular and humoral immune responses. We haven't shown that data. So of the over 20 years I've been working in lipid nanoparticles, I think I'm more excited than I've ever been about this field. I do dream about lipid nanoparticles and their structure and how to improve them. I know that's a maybe I don't want to admit that, but I've been working in this area over 20 years, so I can't help but dream about permutations and the science. It's super exciting, both for genomic vaccines, but also for gene therapy and cell therapy. So I want to thank everybody for listening to me today and the time, and some acknowledgements here as well to the great team at P&I, our collaborators, and our funding agency. So thank you very much.
Yeah, thank you very much, Lloyd. It's indeed interesting to see how, let's say, all the screening and the marketed products have changed the world. I think this was not so much expected a few years ago, and it's wonderful to see that there's more potential. Do we have any questions? So I'm checking, it seems not to be very popular to use the question box. So let's have open discussion later on in the debate room. Good, then we can proceed. Now we can switch over to the next speaker. This is going to be Boris Engels. He's Associate Director at the Novartis Institutes for Biomedical Research in NIBR in Arlington, USA, to talk about the T-Charge, the next generation CART platform with first-in-human data. So the stage is yours, Boris.
Thank you very much. Do you hear and see my screens? Hear me, see my screens?
Yes.
Wonderful. Okay, so thank you very much to the organizers for the opportunity to present our new manufacturing platform that we have established for the chimeric antigen receptor T-cell therapies. And continuing on Lloyd's presentation, of genetic modification. So in our case, we're actually using lentiviral vectors to redirect the T cells and have them attack the cancer cells. So let me see. Okay. Yeah, as mentioned, I'm a full-time employee at Novartis. Novartis has been really pioneering the chimeric antigen receptor T-cell therapies. 10 years ago, Novartis started a collaboration with the University of Pennsylvania after early academic publications showing the efficacy of these CD19 targeting CAR T cells in ALL and CLL. And 5 years later, we had the first CAR T cell therapy approved in pediatric patients with ALL. And then the year later, in 2018, Kymriah was the first CAR T to be approved in a second indication, which was DLBCL. And then in the time in between, we have been able to expand the use of Kymriah and the approvals throughout the world in many countries by now. And then last year, we actually submitted a BLA for a third indication, which is follicular lymphoma for Kymriah. And we were able to present the first in human data of 2 of our new CAR T therapies developed in Novartis, and I want to focus on those in today's presentation. But just today, I actually had to change my slides because we actually got the European Commission approval for Kymriah in follicular lymphoma as of today. So that was a great addition to the slide deck. Having been in the clinic with Kymriah has really allowed us to identify the next steps that we want to include and how to optimize CAR T therapy. And they can be broken down into 3 areas. One is T cell intrinsic factors, where we have seen that the T cell differentiation, their metabolic profile, and also earlier lines of treatment of these patients impact the CAR T product. Also, there are disease factors that play a role, the driver mutations, pre-existing or induced antigen levels in the cancers, as well as the overall tumor burden. And then lastly, the apheresis. So the way that the T cells are harvested from these patients, the timely aspects thereof, and then the cryopreservation, and also the culture conditions as we manufacture the CAR Ts. And again, in today's talk, I'll focus on the T cell intrinsic factors as well as the modulation of the process of generating these CAR Ts. Now, the next generation of platform of generating CAR Ts that we termed T-Charge is based on the scientific rationale that T cells, as they differentiate in natural immune responses, they start off with being naive or T stem cell memory-like. These cells have cell renewal capability, they have a long survival and also proliferative potential. But as the body generates an immune response, T cells will differentiate to central memory, effector memory, and effector cells, where then they gain effector functions such as direct killing, cytokine secretion, but at the same time, they lose their proliferative capacity as well as their survival. So how can we make use of this biology and also the knowledge that if we culture T cells in vitro, they go through a very similar process? That's why for the T-Charge platform, we wanted to retain those naive and stem cell memory cells as well as possible. Now, there are two CAR T programs that are currently in phase 1 trials that are based on this T-Charge process. One is the drug called YTB323, which is an autologous CD19-directed CAR T therapy. This is in a phase 1 trial for diffuse large B-cell lymphoma as well as adult ALL. The interesting part here is that the CAR that we use in this drug, which has a murine binder for CD19, and then these human signaling and structural domains, is identical to the one that we use for Kymriah or Tisagen-Leclucel, as well as the virus that we use in this process, identical to Kymriah. That really allows us to do a comparison and the impact of the manufacturing process on these cells, because the CAR T is identical. Now, a second CAR that is in the clinic is called PHE885, which targets BCMA, which is a target known to be overexpressed in late-stage plasma cells, and is a tumor-associated antigen for multiple myeloma as well. Now, in this case, this is a new CAR T that we developed. So it's a fully human single-chain AV binding to BCMA that was identified and optimized at Novartis, as well as the same human-based signaling domains that we have here. And for today, I'll be focusing on some of the preclinical data as well as early clinical data of YTB323. Now, as I mentioned, for the T-Charge process, we wanted to retain those stem cell-like cells as much as possible. For that reason, the processing time for the T-Charge CAR T cells is limited to less than 2 days. You may have heard that for traditionally manufactured cells, such as Kymriah, the culture times are 9 to 10 or 11 days, in which time these cells differentiate and expand. Now, for T-Charge, we really want to diminish that time in culture, retaining those stem-like cells in the product, and thereby having the potential of infusing much less cells into the patients, but still see the same in vivo expansion as well as efficacy. And shown on the right-hand side here, this is a flow cytometry analysis of the YTB product as well as the Kymriah as a comparator. And if we look at the upper left quadrant, these are cells that express these two markers, express CCR7, but are negative for CD45RO. And these are the naive cells. And you see in the T-charge process, this is the majority of the cells that retain this naive phenotype. If we compare that to a Kymriah product, we see that the majority of the cells are in the upper right quadrant, and these are the cells that are identified as central memory and further differentiated. Now, when we tested these cells in preclinical experiments, these are xenograft models where immune deficient mice were grafted with an ALL tumor. After establishment of the tumor, these mice were treated with different doses of either Kymriah on the left and YTB323 on the right-hand side. And at high doses, both Kymriah as well as the YTB product were able to completely eradicate the tumor, as you see by the red line here. Even when we titrated down, we still see full eradication. But once we hit numbers of below 1 million, we see that 0.2 million of Kymriah was not able to control the tumors, while 0.1 million of YTB was still able, with a delay, but still able to control these tumors in the animals. In addition to the enhanced anti-tumor efficacy, we were able to look at the proliferative potential of these cells in the patients. And we did see that for the traditional manufacturing, we reached a maximum level of a few dozen cells per microliter of blood, while for the YTB323, this expansion was much greater and continued to expand, really showing the proliferative potential that we have for these the cell product. So based on this positive data, we designed this phase 1 trial, the first in human trial, in patients with either DLBCL or adult ALL. The setup is very similar to other CAR T therapies where you have enrollment of these patients. You have the option of bridging chemotherapy up until the infusion of the T-charge product. And one point that I wanted to highlight here is the different doses that we have been using. So this is a dose escalation study, starting with a very low dose of 2.5 million CAR T cells per patient, and then going up to 12.5 or 40 million. And as a reference, Kymriah is dosed as a median of 300, and that is a dose level that is seen for several traditionally manufactured CAR T cells. This is the demographics of these patients. Again, I'll I think skip over some of the details, but the median lines of prior therapy are 2 or more. So these patients have been heavily pretreated before they received the YTB drug. And then looking at the process first. So this is a comparison of the leukapheresis, so that's the T cells that we derive from the patients, as well as the cell product by bulk RNA-seq. We see that we were largely able to retain the stemness of these patients, of these T cells, as well as the memory status of these cells, so supporting the data that we had seen preclinically. Also, we were able to see that these cells were indeed able to expand dramatically in these patients. So what I show on the right-hand side, this is analysis of the circulating CAR T cells, either by flow cytometry on the top or by qPCR on the bottom. The gray lines on the back are reference expansions that we have seen for Kymriah in the Juliet DLBCL trial. And the lines in color are the medians that we see either for dose level 1 and then up to dose level 2. And what was really encouraging to see that for dose level 2, roughly 25-fold lower than the median for Kymriah, we did see expansion similar or even a little bit higher than we had seen for Kymriah. Another detail to point out is that the maximum expansion was reached about 1 week later than we had seen for Kymriah, and that really supports the hypothesis of less differentiated cells being given to the patients, which then differentiate and expand in these patients. And then I want to conclude the clinical data with this the swimmer plot, looking at the responses in dose level 1 in blue and dose level 2 in magenta or pink. So these were the first 20 patients treated with YTB. And if we focus on the dose level 2, we see a really promising response at month 3 of 73%. So 11 of the 15 patients dosed at dose level 2 were at complete response with YTB at this time point, and many of these are ongoing, and obviously we're monitoring these closely. When it comes to the safety, you may have heard that cytokine release syndrome is one of the major adverse events in CAR T therapy, and the safety profile was very favorable, with only one patient so far having a grade 4 or higher, or grade 4 CRS, and 2 patients having a grade 3 ICANS. So closing with this really encouraging data, I wanted to show the current pipeline that we have at Novartis. Obviously, a lot still based on Kymriah, and as of today, the third indication was approved for the use thereof. But we are really focusing on the new manufacturing process, because in addition to the scientific or biologic advantages that I have shown you today, we think that the T-charge process also has the potential to dramatically change the treatment landscape. Number one, as we will have a much faster turnaround of these drugs, and possibly also changing the need for bridging therapies, even in the commercial setting, but also making this more easily manufacturable and accessible. So currently we have 2 CAR Ts on this platform, which is the YTB323 targeting CD19 and the BCMA, and we're really expanding the pipeline to base this on the T-charge. So I'm ending with that. I want to thank all of the collaborators across Novartis, and also all the patients and their families that have taken part in our clinical trials.
Thank you very much, Boris, for this very interesting talk, how CAR T-cell therapies can further evolve with some promising clinical readouts. Let's see whether we have any written submitted questions in our window. Otherwise, I would like to invite you to open the chat box of this stream, where you find a link to that debate lounge. Can you see this, or does anyone have questions about finding it?
Yep, I'm seeing it. It's asking us to join a new meeting, so it looks like I'm getting it here. So I will see you all there.
Yeah, see you there.
Right.
Bye-bye.
Bye.